Welcome back, ladies and gentlemen. Today we're going to explore Chapter 9 and Part A, looking at how genes and genomes evolve. Very fascinating stuff. So let's look at ourselves, human beings, right? Human beings are a single species. We can interbreed with each other. We can have viable offspring. So we are, by definition, one species. Now, how much of our DNA would we have in common? Well, the answer is 99.9%, or by some estimates, 99.8%. So 0.1 or 0.2% of our DNA differs. And those differences are enough to make us identifiable, to give us our personalities and our particular characteristics. So that tells you that DNA must be an enormous molecule. The amount of DNA inside our cells is absolutely enormous. A typical diploid cell has close to 6.4 billion base pairs of DNA. Now remember, we don't just have DNA in our nucleus, we also have DNA in our mitochondria. And the mitochondrial DNA, in the vast majority of the cases, is inherited from our mothers through the egg, right? So as you know, the egg has a much greater volume than the sperm does. Basically, the sperm just delivers a package of chromosomes. Every now and again, a mitochondria may sneak in there, but those are very rare events, maybe 1 in 100,000 by some estimates. So mitochondrial DNA is more stable and therefore used to relate the lineage of humans back in time more accurately than using genomic DNA. And by some estimates, if you go back 5 million years, you'll end up with the common ancestor between humans and chimpanzees. Since that time, the human species has lived a pretty dramatic lifestyle, right? At one point, we estimate about 2 million years ago, there were just about maybe a handful of individuals remaining in our species, and we were on the brink of extinction. We know that from mitochondrial DNA. By tracing back the differences in our mitochondria and using a molecular clock, we can go back to a single female that we believe is the mother of all of us that are alive today. And we call her mitochondrial Eve. And in a similar vein, other scientists have now proposed that we have a single father about a similar time back in our lineage, who's responsible for all of us, right? So our Y chromosome maybe has descended from him. So all these fascinating things, all these fascinating stories are hidden inside our DNA. So the DNA really is a time machine, a time capsule that when tapped into properly can reveal the past. The most important thing when it comes to this particular chapter is, of course, traits, right? So we see here that the blue eye trait evolved randomly in a population of our ancestors as a mutation. So the blue eye gene color, the color of the iris that's blue, is lacking pigmentation. That's the default color of the tissue in your eyes. But for most of us, we have a normal gene that makes pigment, melanin, the same pigment that we make in our skin. And that pigment can be deposited in the iris, giving us brown or black eyes. So mutations can cause us to vary. And likewise, blonde hair is also a mutation from the brown-black hair that our ancestors had normally in the population, okay? And so these genetic mutations can be traced back based on their frequency and the number of base pairs of DNA that have altered. So for blue eyes, we think that that mutation occurred about 6,000 to 10,000 years ago. And before that particular time, blue eyes, if they did exist, were just transient. One person had them, and maybe that person didn't survive to pass them on. So we have all this kind of information at our hands these days, right? So how many different ways can mutation work its magic, right? So mutation is a term that has to be applied correctly. When we scientists talk about mutation, it means a permanent change to the information carried by the nucleotides of the DNA. So the A, the C, the G, and the T is where the information is carried in the nucleotides, right? So when that information is changed, an A is changed to a C, then that's a mutation. Now, there's a small period of time, as you know, after DNA replication, before the replication machine comes by and repairs the damage. At that point, it's called an error, a DNA error. So if the error is repaired, then there will be no mutation. But if the replication machine leaves an error behind and the DNA repair systems don't find that error, and then it's copied into the next round of DNA, then it becomes a mutation. Right? So mutations are permanent changes to the DNA. And they can range from a single nucleotide that's been changed, an A to a T, to the entire set of chromosomes being duplicated. So in certain plants and amphibians, instead of finding 2 sets of chromosomes, you'd find 4 sets of chromosomes or 5 sets of chromosomes, right? So the full range, they're all considered mutations, from a single nucleotide all the way up to entire chromosome sets. In our circumstance, we just need to know the different mechanisms by which the information in our ancestors can be changed during inheritance. So the most common way is mutation within a gene. So here's a gene, and that gene may code for RNA, it may code for messenger RNA, tRNA or microRNA or siRNA, any type of RNA, and it can sustain a mutation. So that's mutation within a gene, right? And that will then alter the RNA, and if that RNA is going to be translated to protein, it may alter the protein. Most students forget that any part of the DNA can be subject to this type of alteration because the entire chromosome is made up of exactly the same molecule, whether it's inside a gene or outside a gene, right? So remember that. So the regulatory part of the gene, which is also made of A, C's, G's, and T's, that can sustain a mutation. So you're going to have a mutation in the regulatory gene. Okay, you can see it right there. What else can happen? What other types of mutation can take place? Another common mechanism that exists in individuals, organisms, is gene duplication. So doing certain genetic maneuvers where the chromosomes are lining up side by side, or when transposable elements are doing their dirty deeds, gene duplication can take place. So here we have a single gene making a protein, and after the mutation, we now have 2 regions of the chromosome that are now making the same protein. And this is great because the original gene can continue to make the protein, and the copy is now able to be mutated at a higher rate, and nature can play with that to produce something slightly different or completely different, right? And we have evidence of that in our DNA. The next mechanism of mutation is called exon shuffling. Shuffling, just like shuffling a pack of cards, right? So the introns in this case are these pink areas. And if you get some type of crossover between those regions, you can have 2 genes lying side by side. And because of a crossover, you now have 2 hybrid genes. So the top one now contains an exon from gene A and an exon from gene B. And the reciprocal of that is an exon from gene B and an exon from gene A. So that will now lead to the production of novel new types of products, RNA or proteins. We spoke about transposable elements. They can move around our genomes under certain circumstances. And if they happen to move, they can move into a coding region of a gene, or they can go into the regulatory region of a gene, right? And in this case, the transposable element has migrated itself into the regulatory component of this particular gene, and that will now interfere in some way by either upregulating, increasing, or downregulating, decreasing the expression of that gene. And there's one last mechanism that we higher eukaryotes don't experience. So you can't do this, I can't do this. But prokaryotes do it regularly. And some very low-level eukaryotes, like yeasts, can do this too, right? So here, instead of having a progeny, I was going to say a baby, a progeny, a daughter cell, or a offspring, here the DNA is transferred between cells horizontally to their neighbors, right, by contact. So here we have two bacterial cells, and they can make contact with each other and transfer the DNA to an unrelated individual, which is amazing, right? And this is the scourge of hospitals, with antibiotic resistance spreading amongst different strains of bacteria in hospitals. So that's called horizontal gene transfer. And that, like I said, is not present in humans as far as we know. So these are some of the mechanisms, just a few of the mechanisms that can lead to a variation and then a change, an evolution in the genome of individuals and then species. Okay, so these slides are simply amplifications of that previous figure. So as well as remembering which cells do what, prokaryotes versus eukaryotes, we also need to remember which organisms perform cellular duplications and produce progeny through sexual or asexual means, right? So the two mechanisms are entirely different. Asexual means that a single cell can clone itself into two cells, and that's called asexual. It doesn't need a partner. But in sexual reproduction, you need another cell from another individual that will contribute half the DNA, as in the case of us humans, right? So the father contributes the sperm, which carries half the DNA, and the mother contributes the egg, which carries half her DNA. And when the two unite upon fertilization, we restore the 46 chromosomes back again in the baby. And then when the baby has its own baby in the future, it will then produce its own gametes that contain half the number of chromosomes, 23 individual chromosomes, right? So understanding the type of reproduction is important in order to apply the right type of biology, okay? Sexually reproducing organisms, as it says here, they only pass on the DNA which is present inside their gametes, right? And when that individual grows up, it will then only pass on its gametes to the next generation. So we have this germline cells. We call them germline, right? Because they are part of your ancestors and your descendants. And those are the most important cells in your body, because the rest of your body is made of somatic cells, right? They're just your cells. So if you cut your finger and you damage these cells, that's not going to impact your offspring. But if you have some type of mutation in the testes or the ovary, and then that can impact the production of egg and sperm. So it's important that we understand these minor differences, right? So in that context, we have this figure which shows this relationship. You can see early on, after fertilization, the zygote, the fertilized egg, begins to divide. And a couple of weeks into development, some of the cells are sequestered, are kept to the side. And those cells will become the germline. They'll become your future sperm or eggs, depending on your sex, right? And the rest of your body will get the rest of the cells, and those are your somatic cells, okay? So only the germline cells are the ones that we are really interested in when it comes to inheritance. And then when you produce your eggs, if you're a female, then the sperm of your partner will fuse through fertilization, and then you'll have your baby here, and the baby will then develop its own similar pattern of segregation of germline cells away from somatic cells. And this is how human generations are produced. And this applies to all sexually reproducing organisms, plants, animals, and many types of protists. Now, when it comes to mutations, there are mutations that are personal to you, as in this case with B. So there's a mutation, and then you get some different cells, and these could be cancer cells. So that cancer is just yours. Or you could develop a mutation in these cells too, because they carry their own DNA, and that DNA can become mutated. And if you get a mutation here, then that will be passed not just into your children, but also into your grandchildren and your great-grandchildren. And then your child will inherit in every single cell in its body that mutation, depending on also what the partner gives, right? So then it becomes a battle between the chromosomes that the second parent gives versus the first parent. But you can see here. So germ cell manipulations are very much controlled by the scientific community because of the dangers that they pose to the human species, right? They can have a dramatic effect, not just on you, but future generations. So point mutations are the smallest level of mutation that we know of, where a single base on the DNA can be changed. When can that happen? That normally happens during DNA replication, when you're copying the DNA so that you can give it to the daughter cells, either through mitosis or meiosis. And as we said earlier, DNA is DNA. The entire chromosome is made of DNA, right? There's no part that's special. So the telomeres have DNA, the centromeres are DNA, and the coding regions and the non-coding regions, the euchromatin and the heterochromatin, the tightly packed and the loosely packed DNA, all that is the same exact double helix. Every part of the DNA molecule is subject to mutation. In that sense, then, we can divide the zones of DNA into coding regions that code for proteins and regulatory regions that regulate the behavior of that particular gene or gene set. So as you can see here, some people are lucky, right? They have mutations that don't affect them drastically, but they do prevent them from picking up certain types of diseases. So the malaria parasite, Plasmodium, which is a protist, belongs to the kingdom Protista, it binds to red blood cells by using a protein that's on the surface of red blood cells. So it has to find that protein, latch onto the cell, and then enter the cell. In some individuals, that protein is defective because of a mutation. In this case, the mutation is in the regulatory part of the gene. Therefore, the protein is so defective that it's not made. So it doesn't appear on the surface of red blood cells. So if it's missing from your red blood cells and the parasite enters your bloodstream, the parasite cannot latch onto your red blood cells and therefore replicate and cause malaria. So you will be resistant to malaria, which is a fantastic thing. But what are the consequences of this particular protein missing on your red blood cells? Well, it doesn't seem to have a major consequence that we know of, right? So in this case, it may be a good thing. The next example is quite fantastic. Most people can relate to this. We're going to talk about lactose intolerance. Simply put, the enzyme lactase is responsible for starting off the digestion of the milk sugar carbohydrate called lactose. So lactase is an enzyme that binds to lactose and cuts it into two monosaccharides. And that's the beginning of the digestion of that particular sugar. Now, under historical conditions, we only had to digest milk from our mothers. By the time we were weaned off the breasts of our mothers, there was no need to go back to milk because we did not live around animals that gave us milk because our mothers would stop producing milk once we were weaned. Therefore, it didn't make sense for our bodies to continue to make lactase after maybe the age of 5 to save energy. So under normal conditions, most people are lactose intolerant, except in 2 parts of the world. In these parts of the world, which are Northern Europe and Africa, our ancestors began to live in communities where they farmed and they had dairy cattle. So they kept cattle as a supplement to their diet. And these communities, they drank milk. Now, the milk didn't cause the mutation. The mutation was a natural event. But over time, as more and more people were born with this ability to produce lactase into adulthood, these people were able to drink cow's milk. The reason why they could do that, there was a mutation that prevented the gene from being turned off at 5 years of age. And that mutation, either you had it or you didn't. If you didn't have the mutation, then you stopped producing lactase when you reach 5 or 6. And if there was a famine in the future and the only thing available was cow's milk, then you would get bloating, diarrhea, and most probably you would die from those ailments. And therefore that mutation, that wild type condition would go away with you. And those people that could drink, just by chance, cow's milk, they were able to survive the famine and then move on. Therefore, the incidence of this particular mutation increased in these two populations, Northern Europe and Africa. So if your descendants come from there, there's a very high likelihood that you are not lactose intolerant. But if you come from the American Indian, the Incas, the Maya population, i.e. Central and South America, or you come from Central Asia or the Far East or Southern Africa, right? Then you can see over here, you have a very high likelihood of being lactose intolerant. So the more yellow the color, that community, those communities don't have this mutation, and therefore they can't drink the milk. This particular slide wishes to educate us on a similar concept as the lactose-lactase system. Here we can see that certain genes are responsible for activating other genes, so they're called master genes. And the activation of these master genes leads to the production of certain body components. So these master genes are responsible, for instance, for producing an eye. So if you turn off the master gene at a certain point in the development of a human fetus, then eyes will not develop. Or if you put an activation of a gene in a different part of the body, then you could get eyes on the back of the head, right? So this is how the body works. It's a very modular design, similar to what we consider IKEA and their furniture. In this particular example, we have an ancestor that had a normal-sized tail, a normal-sized snout, and the interactions were straightforward. But because of a mutation in this column here, the mutation has changed the interaction of one gene with other genes. For instance, in this case, gene 1 is producing a protein. And that protein is going to become a control element for other genes. So this protein here will bind to gene 3 and to gene 2. And it was supposed to activate both genes in the past. But for some reason, there's been some type of change. And now you can see one of the genes is turned off and the other gene continues to work. And by turning on this master gene and producing this master protein, that will then bind to many other proteins, and that will lead to a long tail and a long snout. Now, a different mutation in a different gene will give rise to an alternative where gene 3 is no longer producing its product, but gene 2 does. Now, these proteins, these master proteins, will then have their effect on different genes in a different way. And that will result in a shorter snout and a shorter tail. So mutations in regulatory parts of genes can affect the expression of their gene. And those genes may be important in controlling other genes. So you get this domino effect. And this is how we can explain the existence of different types of structures and biochemistry in different individual species that are closely related. Mutations like this can give rise. For instance, if you look at the chimpanzee, it's got hair over its body. Whereas if you look at you and I, we don't have any hair. So we can explain that by a single mutation that sort of cuts off the production of hair at a very early age during our development, before the hair even appears on our skin. Whereas in a chimpanzee, that gene will be activated for all of its life, and the chimpanzee will be hairy until the day that it passes. The next segment of the textbook talks about mutations and measuring the rate of mutation. If you have too many mutations, then the chances are that too much of your biochemistry will be disrupted and that you will not be able to lead a normal life, particularly reproduction, right? So you won't be able to reach maturity to produce babies of your own. Therefore, natural selection will kick in and you will then take your genes to your grave. If there's too little mutation, then there's not enough adaptation to the environment. So our babies don't have enough variety to cope with future challenges. So mutation has settled down using the Goldilocks principle, where there's just enough mutation to satisfy both equations, and not to disrupt your biochemistry, but to give enough so that natural selection can take place. So mutation rates can be measured. And how do we measure mutation rates? We can use certain experiments, right? So the mutation rate in bacteria for certain regions of the chromosome varies. So some parts of the chromosome mutate at a higher rate than other parts. They're called hotspots. But let's not look at that. So the rate of mutation in E. coli has been calculated, 1 in a billion nucleotides copied, right? So how can we determine this? Experiments like this, where bacteria that were already mutant were mixed together in a flask and grown to huge quantities, billions and billions. So that increases the probability that rare events can be detected, right? If you had 10 humans and the mutation rate was 1 in 10 million, then you would never be able to detect that frequently the rare mutation in 10 humans. But with bacteria, because they grow to enormous quantities and they can do that overnight, we can then detect rare events. So in this particular experiment, a single strain of bacteria was grown in the lab and it was made into a mutant and then confirmed. And the mutation is in the histidine gene. What is histidine? It's an amino acid. It's a vital amino acid, right? Under normal circumstances, a bacteria will die if it doesn't have histidine supplied to it from the outside. So what the scientists do, they give these bacteria in this flask histidine to keep them alive and growing. But if they didn't put histidine in the flask, the bacteria would be all dead, right? Because they have this mutation inside the gene for making an enzyme in the histidine pathway. Okay. So what the scientists want to do now is to grow the bacteria to enormous levels so that mutations are likely. And mutations are sporadic. They happen randomly up and down the DNA. We're looking for a mutation at this precise spot that will change the AT that you see there back to what it was in the normal bacteria, the ones that can make their own histidine. And that is given over here on this side, right? So the GC base pair is the normal wild type, and that will allow that gene to go back to normal. So what we're looking for is how many bacteria out of this broth of 10 billion bacteria will be able to randomly, just by chance, grow when they're put on a plate that doesn't have any histidine. Those bacteria that are not fortunate enough to get the mutation to revert this back, to convert this back to wild type, they won't be able to grow. So if you get a mutation in the first AT, that's not good enough because you're not restoring the AT on the end back to what it should be, the GC. If you have a mutation over here in the yellow, that's not good enough either. Any mutation anywhere along this chromosome is not good enough. You must have this one go back to exactly what it should be with a G on top and a C on the bottom, right? And you can see here, because we know the density of bacteria in the flask, we can measure how many colonies grow on the plate. And that will give us an estimate of the rate of mutation in this flask. And by putting different chemicals in the flask, like toxins or plastics or makeup or food molecules or dyes, anything you like, you can then measure the rate of mutation. Moving on to the next slide, here we see one mechanism that can lead to gene duplication, where you have one gene initially, and then after the mutation, you have two genes on the DNA. So the most common mechanism that leads to gene duplication is when the chromosomes line up wrong. This is how they're supposed to line up during a certain phase of the cell cycle, right? They're supposed to line up identically, side by side. But sometimes repetitive DNA, as you can see here in red, that repetitive DNA causes the cell to become confused. And the chromosomes line up in this fashion here, where the repetitive DNA is used to match the pairing. But the genes are now staggered. And if you get any kind of exchange of DNA in this repetitive region, then you will end up with one chromosome that has a copy of the gene, where the DNA will flip over to the second chromosome. And then you have a chromosome that has the opposite, which is that small section there flipping down to this section. And there's the short chromosome, right? So when this chromosome is inherited, the top one here, by a daughter cell, that daughter cell will have two copies of the gene. Whereas this chromosome here, when that goes into the other daughter cell, the chances are that daughter cell won't survive, because this gene may be very important, and that daughter cell is lacking that particular gene, right? But that's enough. This daughter cell that gets the top chromosome will then reproduce in the future, and every time it reproduces to have its own offspring, its own daughter cells, they will carry a duplicate of this particular gene. So that's how gene duplications happen. What are the consequences of this? Well, the most important consequences are that it can change the way that proteins can interact. So you can see here, this particular polypeptide, it's a single polypeptide. It has a one beginning and one end, right? And it folds to form these secondary structures. So this is a single molecule of hemoglobin. And this is what we find in early animals, our ancestors, right? And lower down the food chain, lower down the evolutionary tree. But because of gene duplication, we now have hemoglobin that looks like this. And this hemoglobin is more useful at carrying oxygen. It's more clever at carrying oxygen than this type. So this gives an advantage that we can now get larger bodies that can carry more oxygen from our lungs to our tissues compared to this. So thank God that in our past, a long time ago, there was gene duplication. And gene duplication can now be measured because of our technology. So if you look at part of chromosome 11, human chromosome 11, we have the beta globin gene cluster. What does that mean? It means we have 5 genes who are very similar to each other, but they perform slightly different functions, right? Gene epsilon, that's made in the embryo, right? And then we have gene delta, that's made in the baby, and early postnatal life. And then we have the beta form, that's the adult form of the hemoglobin beta chain, right? So we can follow back in evolutionary time, and these are millions of years into our past. We can follow back when we know that these duplication events took place, okay? So these two are broken genes. These are pseudo genes. They don't function well. And you can see here how they were once related to the alpha globin genes, which are the other half of the hemoglobin molecule with the 4 subunits on the previous slide. Let's just show you that. So 2 of these are alpha, and 2 of these are beta, right? So the beta come from this gene in the adults, and the alphas come from their own family tree, which is not shown here. But thank God there were gene duplications 500 million years ago, because otherwise we would just have a single globin gene that was only limited in carrying oxygen around our bodies. The next slide just proves to us that you can get a drastic change in the number of chromosomes between related species. So this is Xenopus, and the Xenopus frog has a cousin that has more DNA than it does. And the reason it has more DNA per cell, twice as much, is because it had a mutation where the DNA was duplicated by a mechanism that we don't discuss in this class, right? And you can see if that toad, or that frog, is twice the size of its cousin. Is that always the case? Not always the case, but there's a general trend. The bigger your genome, right, in terms of copies compared to your ancestors, the larger you are. So the present day crops that we consume for making food, wheat, barley, oats, etc., rice, they are multiples of their ancestors. So maybe the ancestor had 2 sets of chromosomes, but today barley will have 8 sets of chromosomes, right? And that makes that particular plant larger and more useful for us because we have more food per plant. We have evidence that in simpler organisms and our ancestors, genes were smaller. They normally consisted of a single domain. So a domain is a functional part of a protein, right? So that domain could bind to DNA. Say the green here binds to DNA, right? Or they could perform an enzymatic function. So chymotrypsin. But then because of gene exon shuffling and gene duplication, we ended up with new combinations of these modular proteins. So this protein here has been constructed by exon-intron exchange, as we mentioned earlier. And now it is a compound protein. It consists of 1 unit of that, plus a new unit, and plus a unit of this. And indeed, we do find within our bodies right now, proteins that are duplicates of each module, plus bits and pieces of other modules. So nature likes to tinker, not invent something new. So you don't see anything red on this slide because nothing new has been invented. All nature does is take existing components and then shuffle them around to make new patterns and new proteins. Okay, so that's what's going on here. So the last example is this, just to prove to us that the way that nature works is through a modular design, an IKEA type design. Scientists have been able to capture in the wild or produce naturally in the laboratory using mutation some weird monsters. So in this case, what they've done is they've found flies, or they've produced flies themselves, either way, in the laboratory that have legs on their antenna. They have legs on their antenna. How can you do that? Well, under natural conditions, transposons, these selfish pieces of DNA, they can move around. And if they move around into a place where they turn on a gene that shouldn't be on, a master gene, then things can go wrong. So in this particular case, the transposable element has moved in the head of this creature into the master gene for making legs, and it's turned on the legs in the antenna. So the body will obey whatever the genes tell it, and therefore that master gene turns on the development of legs. And you can have many other types of mutations where you have double sets of wings, or you have eyes on the legs, right? So that tells us that animals and Mother Nature have decided to use modular design with master genes activating submasters and submasters activating individual genes in turning things off. So your arms and your legs are made of a master gene being turned on in those 4 locations. Therefore, you have 2 arms and 2 legs. But if you turned it on up and down your thorax and your abdomen, then you would have as many legs as a millipede. And in fact, that's what millipedes are. They're just genes that have been duplicated and turned on different segments of the body. Let's have a look at the bottom part of the slide. It says that factor VIII mutation, which results in a bleeding problem, that did not exist in Queen Victoria's family or Prince Albert, her husband's family, right? But it did exist in some of her children and their descendants. So how do we explain this? Well, it seems that in her germline, in the ovaries of Queen Victoria, maybe a transposable element, an allo sequence, moved. And it moved and disrupted the functioning of one of her chromosomes with factor VIII. And any child that inherited that chromosome through her ovary was then impacted. And because it's on one of the sex chromosomes, the X chromosome, it's mainly going to affect male offspring. Because males only have one X chromosome, and they get their father's Y chromosome, whereas females get an X from each parent, right? And because it's a recessive mutation, it's going to then impact males more than females. So again, we have evidence to support what we said at the beginning of this chapter, that mutations come in all shapes and sizes. Thank you.